So I should have said this the very first day I started, but in the materials that I submitted that are online at the website are the handouts for these lectures. So some of you have laptops, and if you wanted to look at the PDF as you're going, or some of the stuff is in there. So this is lecture 3 under my materials. So maybe that's helpful. I don't know. I'll write mostly on the board. Maybe there'll be a little bit of show a figure once in a while on the PDF and point to it, but I'll write on the board. So on tomorrow in the afternoon, I'm going to give a talk, a research talk, and I'm going to link together the models that I presented the last 2 lectures, looking at how we take a structured population, with models of how phenotype evolves. In the last 2 lectures, I linked the demographic life history models to change in allele frequency. And today we're going to focus on how a phenotype changes and how the underlying alleles that control that phenotype might change, with the idea that then at the research talk, I'll maybe put it all together somehow. It'll all work. All right. So let's first just think about an observation. So when scientists look at populations of real organisms and look at a trait they're interested, some phenotype they're interested, they often see, not always, but often see a Gaussian distribution of phenotypes. At least, depending, sometimes you have to plot the phenotype on a log scale, but often you see a Gaussian distribution. So it's not so surprising. So why is that not surprising? Why do we see Gaussians all over the place in life? Central limit theorem, of course. So if we have some variable that is determined by the sum of a bunch of independent variables, we're going to get a Gaussian distribution. So sort of at the heart of some of this is the idea that the phenotype we're looking at, and right now we'll consider a single phenotype, but later we'll look at phenotypes that are complicated, that have many different dimensions. Underneath that phenotype is a lot of genotypic information that leads to that phenotype. And the simplest case is when it's independent, but it doesn't have to be. So we have in sort of the same, so Daniel was talking about earlier having a high dimensionality space underlying some phenotype. In quantitative genetics, there's a high dimensionality space too, but it's a genetic space that's underlying phenotype. So we have some distribution of phenotype, and we want to think about how it changes. So the simplest possible observation we can make is say, what if there's some kind of selection on this phenotype? And the easiest one to draw on a board is truncation selection, which is just basically saying that at some point t of phenotype, I draw a line, and I say all the organisms who have a phenotype on that side of the line, I allow to reproduce for the next generation, and none of these do. Now, I've drawn this very strong selection, but it doesn't have to be, right? It could be something like that. So truncation selection is the sort of simplest thing to think about. Nothing that I'm going to talk about today depends on it being truncation selection. It's just the easiest thing to sort of go through. So let's just describe what's going to happen, what's happening with phenotype. So in this case, I've changed notation several different times in here, so I'll try to be consistent. In this case, we have some mean in our original population. We'll call that mu. And then we might imagine that if we just consider the organisms that we're allowing to reproduce, they have some mean mu sub s. And for this example, I've chosen things so the mean of the selected individuals is greater than the overall population. It doesn't have to be. So we can define something called a selection differential. And that's just how strong selection is. So if the difference between those two means is very, very big, selection is very strong. If the difference is very small, selection is very weak. Now we imagine then, in the future, these individuals reproducing. I'm not going to draw it underneath here, but that's what's shown up here, the same picture. So here we have this Gaussian again. It's using different notation, that's fine. We've truncated these individuals, and only these individuals are allowed to reproduce, and in the next generation, they're the parents of a new generation. That new generation has a new mean, and I might draw it here, these guys. You know, they have some new mean. We'll call it mu prime. And we can also then define something called the response to selection. Which is the difference between this generation, and the Gaussian is shorter, just because I didn't have as much space, pretend it's the same, and the original population. This is how much selection's changed my phenotype. And we could calculate selection differentials and responses to selection, even if we use something other than truncation selection, but it's a lot easier. So typically, in this case, we're truncating to the right of the distribution. And if you look at this figure up here, the way it's drawn, mu prime is greater than mu. So there is some response. But mu prime is less than mu sub s. It's not as great, the mean isn't as far to the right as the selected individuals. So if we think about why that's true, why the mean of the next generation is greater than the mean of the previous generation, that's because some of those selected parents had favorable genotypes, presumably, that affect this phenotype, and they pass it on to their offspring, and so their offspring have a phenotype that's more close to the truncation point, that's in this case, a higher value. But why doesn't the mean of the next generation, why isn't it as extreme as that of the selected parents? Why is that? Only these are the parents. These are the only parents. Relative. I'm not sure I'm understanding what you're saying. The relative numbers. That this is a larger that this is a bigger number. Yes. Oh. No, that's not necessarily why. So there's two main reasons. One reason is you have to think about why individuals are in here. Some of these individuals are in here because their genotypes are such that they make a phenotype that's that extreme. But some of those individuals are in there, and they may have a genotype that would maybe put them in here, but the environment that they were raised in gives them a phenotype that's over here. So one reason is that not all of the phenotype is due to the genotype. And there's another reason. I should have said this is sexual reproduction, but if I tell you it's sexual reproduction, what's another reason? Yeah, it's all the reasons that children are not exactly like their parents, right? So in sexual reproduction, a random mating population, these parents give alleles, generally, rather than full genotypes. So recombination, and also segregation at a single locus as well, means that favorable combinations either within a locus or across loci get broken up, and so the phenotype that you see might not reflect exactly the parent. All right, so we won't get the exact thing. These two quantities are very famous in quantitative genetics, and they together show up in something sometimes called the breeder's equation. It's called that for the reason you might think, because it's used in animal and plant breeding. So that equation says that the response to selection is given by how strong selection is placed, multiplied by a quantity called h squared. And it's one of those things that, for reasons lost to history, at least I have never figured it out, we never give h. It's not like the square root of this quantity is something that it's always h squared. H squared is sometimes called the realized heritability. It's also sometimes called the narrow sense, if I can spell narrow correctly. And we'll see later that under a couple different models that we can think about, this quantity basically tells us how much of the variation that exists for this phenotype is the kind of genetic variation that can be passed on to offspring. The additive genetic variation, we'll call it. So we're going to talk about different ways of defining this. You can actually calculate a heritability from a simple selection experiment. So you can take a population. I don't know if this is going to show up here. The top is. So this is an image from a textbook. So this is a distribution, sort of Gaussian, of the weight of seed in phaseolus, in beans. And this is, some distribution here. And so the original mean is some 403.5. I think it's milligrams. So mu is 403.5. And in this selection experiment, they placed truncation selection at a point here, t, of 650. And when they looked at the parents, they had a mean of 691.7. And then they let the offspring of those individuals breed, cross, and there you have a new distribution. It's shifted to the right, not as far as this, but it's definitely shifted towards larger seed size, or larger weight. And that value is 609.1. And then with these numbers, you can calculate the heritability. It's the response divided by the selection differential, which in this case is 6091 minus 403.5 over 691.7 minus 403.5. And this ends up being approximately 713. This is saying that approximately, there's about 70% of the change that you applied due to selection showed up in the offspring. And you might ask, is that a high heritability? That's actually a high heritability. There's many traits that show that kind of heritability. There's other ways to calculate heritability, and I will talk about that in a second. Actually, I'll talk about that right now. This is the result of a selection experiment. You can also calculate heritabilities. I won't talk about it too much. By looking at the resemblance between relatives. So that graph at the top shows flower beetles. Oh, see, hoping this would be my big flower beetle moment. So what it shows is the phenotype here is the pupil weight of offspring in micrograms. And these are the offspring. And these are the parent. In this case, it's the father, and they're using the father to try to control for maternal effects. And you see there's quite a spread, but there is a regression that's significant. And in that case, for this particular trait, the regression is 0.11. And I'm not going to go through the derivation, but it can be shown that in the case that we have a trait in one parent, and the same trait measured in the offspring of those parents, that the heritability can be estimated as twice that regression. In that case, it's about 0.22. When I teach my genetics class at the University of Kansas every year, including this year, the very first day of class, I give the students a piece of paper and I say, write down your height. We do it in inches because we're Americans. We have to be annoying in inches. And then if you know it, write down the height of your biological mother and your biological father, if you have that information. So, and it's optional and anonymous, they don't have to do it. They mostly all do it. And then later on we put that together. And at the end of the semester, which will happen in a month or so, I will do a regression like this on, not on the parent, but on what's called the mid-parent. And on the mid-parent, that factor of 2 goes away, and it's a direct estimate of heritability. And I've been doing that for about 20 years. And every time I do it, I don't teach every year, I teach every other year, so genetics every year. So I've done it maybe 10 times. And almost every single time, every single time, I get a heritability estimate for height between about 0.65 and 0.7, I think 7.8 is the highest. Maybe I've gotten 0.8. And my estimates from my genetics class match perfectly those of real studies where they really look at real heritability of humans. Because human height has a heritability of about 0.7, about the same as this bean example. So, so people calculate heritabilities all the time. So if you later on, yes? Yes? It's not exactly. It's what it's giving you, it's actually giving you the ratio. We'll see later. It's giving you it's giving you this relationship between the response, it's the proportion of the selection differential that's the response, obviously. But the interpretation we're going to lead to is it gives you the ratio of the additive genetic variance to the total phenotypic variance. And we'll actually derive that. So, all right. So this is just phenotype. This is just selection and phenotype. There's no genetics here, really. There's sort of an assumption of genetics there. So immediately we're like, well, that's nice, but I'd like to have a model that explains this in terms of that concept I first said. So I said, we have a phenotype, and we think it's made up of many, many, many independent things giving us this Gaussian. Can we come up with a model for that? Well, we don't have to. People did it a long time ago. It's sometimes called the multifactorial model, and it's basically a very simple model about the genetic underpinnings of this. And so what we're going to do is consider a single locus first. Now, it's not because we think only one locus determines these traits, but we're focusing on the effect of a single locus in isolation. In fact, we assume that there's lots of loci affecting the trait. We need that in order to get a nice distribution. Okay, so let's draw another Gaussian. Go over there. Let's draw a single locus model for everything. A prime, A1, A2. So at a single locus, let's just think of 2 possible alleles, A and A prime. And we might ask ourselves, what's the distribution of whatever phenotype, whether it's the weight of the seeds or the height of humans or the pupil weight in the tribolium? What's the distribution for, if I looked at a population that was made up entirely of one genotype? So we might imagine something like this. Over here. And I'm going to warn you, I'm not very good at drawing Gaussians that are really symmetric. So you're going to pretend that they're all beautiful and symmetric, right? And I'm also going to draw these separated by quite a bit. So it's easier for you to see what I'm doing. But really, what we'd like is to think that they're very, very close together. So actually, I don't know if you can see this picture. You can. So here's like how a professional would draw it, really, really close together. Yeah, we're not doing that. That's what I have. So we're going to define the means of these 3 distributions. We'll pretend that's on the mean. And we'll say what's the average or mean phenotype. So we'll call this MAA, or mu-AA perhaps. And I'm going to define it in the following way. And I'm going to say that this one is A prime, A prime. This one's A prime, and this one's AA, just the way I'm drawing it for this case. And then I'm also going to make one other assumption for right now, and that's of Hardy-Weinberg equilibrium. So the frequencies of these three genotypes in my population are given this way, right? And obviously P plus Q is 1. Okay, so why did I write it this way with these coefficients? I want to write all three mean phenotypes in terms of each other, sort of their relationship to each other. And we can sort of see, so M is just a reference value. It's not generally the mean overall. Well, I'll show you where it's the mean. And we're just defining a reference value and asking what are the deviations from that reference value, with the idea that in our minds we have some group of alleles and genes and alleles in those genes, and also environmental effects, all of which add up to a phenotype. And the effect of this particular genotype is to shift that distribution a little bit. So we're going to shift it away. I think you can see that if D is 0, that means no dominance. And we have additivity of the alleles, right? If D is 0, then the heterozygote is exactly intermediate, and adding an A prime moves you this way by amount little a, and adding an A moves you in the other direction, all right? Also, if D equals A, then big A is dominant, completely dominant. If D is equal to little a, negative A prime is dominant, completely dominant, and anything between that is, sort of partial dominance. And so we can, for any particular trait, we could calculate what those are. And let's use an example. I can't remember who someone put a picture up. I think it might have been Joaquim, who put up a picture of Sewell Wright. And I don't know if you remember what it looked like, but he was standing, it's a famous, famous picture. He's standing in front of a chalkboard, like this, and on the desk in front of him, he's holding something. He's holding a guinea pig. And I've always thought that looks like he's about to erase the board with the guinea pig, but I don't know that he actually ever did that. But let's borrow from Sewell Wright for a moment. So you can't see that, but I'll rewrite those numbers. We're going to use an example from Sewell Wright's data. Whoa, that was a mistake. Okay. I didn't do that. We'll just keep going. That's the phenotype. And it has to do with the amount of black fur on the guinea pig. And to get it to be a nice normal distribution, it's actually the arc sine of the square root of the percentage of the coat color, but it doesn't really matter. So there are three genotypes. Sewell Wright used different notation, but we'll use our notation. And so these are the three genotypes at a particular locus that affects coat color. And the means from this example, these are real guinea pigs. Very well. So those are the three values that Sewell Wright found for the three genotypes. And basically what he had is he had, populations, breeding populations of these guinea pigs that were all one. So he could pull out the ones that were the same genotype and measure their phenotype. so we can say, well, little m, is the reference value that is midpoint between the two homozygotes. So m, for this example, has to be halfway between those two. And then d is just the deviation. Well, a is the deviation in one direction, so we can calculate it using either one. So for example, a is, looking at that, we can see that a has to be the mean of, for example, the big A, minus m. Which in this case is 1.202 minus 1.075. 127. And D, similarly, is the mean of the heterozygotes, minus m. And they are 1.0859 minus 1.075. 6, and obviously, minus A is just minus 0.1727. So D in this case is negative 0.016. And we can see just looking at these that from our definitions, right, this is not halfway between the two of them. Right? There is dominance in this case. All right, so this is the way people calculate these, and they assign these amounts. So the key to what we're doing here is we're saying one locus is a focus that we're looking at, and it's shifting these distributions just a little bit. We'd like to have a model for how that's actually happening. So we're going to calculate a few things about in general about this model. So we'd like to know what is the mean value, what is the mean phenotype in the population, given this information, given this sort of general model. Now I said what we were going to assume Hardy-Weinberg, so it's very easy to calculate that. We'll call capital M the mean of the whole population, and it's just given by the relative frequencies of the three genotypes under Hardy-Weinberg and their mean values. And a little bit of algebra gives you this quantity. And one thing you might notice that unless you have equal allele frequencies for this 2 allele case and no dominance, the mean phenotype doesn't usually equal that reference value. It can, but it doesn't. So this writes the mean phenotype of the population in terms of 3 quantities. Our reference value, a value that has to do with A, and sort of a value that has to do with D. And so that lets us start to put an interpretation onto A and D. So A is sometimes called the allelic effect. So the independent effect of either allele A or allele A prime on the mean of this phenotype. And D is what we call the dominance deviation. All right, that's the mean. And that plays a prominent role in sort of quantitative genetics. Now I'm going to find another quantity that's important. The regression coefficient of phenotype onto genotype, but a specific type of genotype, onto the number of one of the 2 alleles, and in this case, it's the number of A alleles, in this case. So we can show, get the regression coefficient, we can first calculate the variance of the alleles. Right, the expected number, we'll call that, what did I call it? I called it X, variance of X. The expected number of A alleles is very straightforward because We have 2 for all of our homozygotes. We have 1 for all of our heterozygotes, and we have 0 for all of our other homozygotes. This ends up being 2p. And we can also find, x squared the same way. This ends up being 4p squared plus 2p cubed. And finally, the variance then ends up being 2pq. Comes out a whole bunch of different ways. Okay, so that's one of the 2 things we need. We also need the phenotype. So we'd like to know the mean phenotype. That's easy. We already found that. So let y equal the phenotype. We already found the mean phenotype over there. So I'll just write m, but this is it here. The covariance between the number of alleles and the phenotype. We can write it out. And that's this quantity. And then finally, we put that together somewhere. And get the covariance between the number of A alleles and phenotype. And a little bit of algebra gives this thing, 2pqa minus 2pq q minus pd. And finally, the regression is that covariance over the variance of x. And that ends up being this quantity. So we're going to see that this expression ends up being very important in defining heritability, which we were talking about before, in terms of the effects on these alleles. So sometimes a drawing is helpful. I have the actual drawing for my friends, the guinea pigs, a sketch of theirs. So that's too small to be useful. I'll draw it over here. What that tells me is if I make a graph, and here I graph mean phenotype, or phenotype. And here I graph the number of A alleles. So the values are 0, for this case 1 and 2, right? I have values for the 3 genotypes. This is the A prime, A prime. This is the A prime. This is AA. That top graph, that's what it is for the guinea pigs. Some values like this. The slope of this is that quantity. So why is that important? It's important because if there was no relationship between the number of A alleles and the phenotype, then it doesn't matter how much selection we apply on this phenotype, we're not going to change the frequencies of these alleles. It's only if there's a relationship between the frequencies of the alleles, and the number of A alleles is a proxy, obviously, for that, and phenotype. So we knew those to be true. So that makes sense. At this point, I want to talk about dominance for just a second. So dominance is actually very important. And I won't go into ridiculous amounts of of detail. But one of the things that dominance does is it decreases heritability. And I can give you an example for the reasons that we were talking about at the beginning. Remember I said, why don't the offspring of the selected parents look as extreme as those selected parents? And one of the things you guys actually said, recombination and segregation, right? Dominance is basically what makes things at a single locus not additive, right? So there's a little drawing here. This is from Hartle and Clark's book, right? This is a little picture where you should think of this maybe as being something like coat color. This is a case where we have two different loci affecting the trait. And in this case, every capital letter, big A or big B, adds 1 unit to the phenotype. And every lowercase letter, either little a or little b, doesn't add anything. And so the possible phenotypes range from 4 to 0. And here, the only thing that's changed is that there's dominance. There's no interaction between the two different loci, there's just dominance. So now the biggest one is still 4, and the smallest is still 0. And I might say, well, I'm going to apply selection on this and ask, sort of like I did with my phaseolus, my seeds, and say, well, what's the realized heritability? If I apply a certain amount of selection, what's my response, and calculate H-score. And if I go ahead and say, I'm going to take the 2 lightest colored sets of guinea pigs or whatever and let them breed. In this case, it's these guys and these guys. And these guys, it's these. When I do that, I get very different. So with additive alleles, for this example, completely additive, and the trait is completely determined by these 2 loci. So in this case, it's kind of the ridiculous toy example, heritability is 1, right? It's complete. Just adding dominance, in this case, drops heritability to 0.81. And that's because when you have dominance, some of the phenotype is due to the interaction between alleles at the same locus. We'll talk about later what happens if you have interactions between alleles at different loci, if you have epistasis. So under sexual reproduction, in general, over time, under sexual reproduction, every generation you lose that relationship between the diploid genotype. Okay, so that's what happens to the phenotype, so to speak, right? Or not what happens to the phenotype. How can we describe the phenotype under this really simple model? So now let's go back to that same example before and say, okay, let's apply selection on the trait. And how do things change? And we're going to look at 2 things. We're going to look at how the mean phenotype changes, and we're going to look at how allele frequencies change. So to do that, we again could do any kind of selection we wanted, but truncation selection on something distributed as Gaussian makes the math really simple and easy to draw on a chalkboard. So we're going to do that. And I'm going to try not to spill water everywhere again. You guys are watching me carefully to see if I do that again. So imagine that we applied truncation selection on a distribution the same way we did before. And we truncate at some place T. We again have some Gaussian distribution. We have some mean here. We call it M. And we truncate at T. And we have some N sub S there. If we allow the height of this curve at this point to be z, then the area here, b, we can get just from the distribution of the Gaussian. And one thing we can do is say, what is the difference between the selected individuals and the overall individuals in terms of the variance of that distribution. And for a normal, that's just z over b. So what we're going to do is look at the phenotypes of the three genotypes separately and ask what proportions of those three distributions, which I erased, they were over there, of those three distributions will be kept when I apply selection. And I'm going to apply truncation selection on the right, and I'm going to use big A as the rightmost. So I'll keep more of big A, and a little bit less of big A, of A prime, and then a little less from that of A prime, right? That's what it'll look like. So if I drew all three of them again, you know, oh, I did a terrible job. And I cut here, you can see that there's a bigger slice of this curve, which is the AA curve. And there's a sort of a smaller slice of the next one, and there's a really small slice of the last one. We'd like to know how big those slices are. Let's just focus on these two curves. And I'm going to try to draw them so it's useful. And I'm exaggerating the difference between the two of them. Like that. I did that correctly. And I'm going to cut here. Here's T. So I have a piece this big of the AAs and a piece that big of the AA primes. And the difference between the two means, and I've exaggerated it quite a bit, this is M plus D, and this is M plus A, and so the distance between the two of them is the difference between that, so it's A minus D. So that's this distance. Imagine then that I shift those two distributions and superimpose them so these two lines are sitting on top of each other. I'm going to imagine sliding them over. So now the two lines are superimposed on each other. They're sitting here like this. How much did I shift this part over here? I shifted it A minus D, right? That's how big this slice was. So now, if I drew this, if I did this carefully, and showed how many of the AAs there are, and how many of the AA primes, here those things are together. Here, they're separated by a distance A minus D. And A minus D then gives the width of this difference, which is the difference in the proportions kept that were AA and AA prime. Now, I drew this like they're really far apart, but an aspect of this model is that each of these alleles shifts the distribution a very small piece. So really, that piece is more like right? It's really a very small piece. It's still A minus D, but it's very small. Well, since it's very small, I can say that the area of that piece is approximately the height of the Gaussian at that point multiplied by A minus D, right? So that area is A minus D times Z, which is the height of the Gaussian at that truncation point. So that lets us define the difference in fitness for these two genotypes. So I'm going to call the fitness of AA W11 and the fitness of AA prime W12. I should have used ones and twos, that's okay. And it's approximately Z times A minus D. And I can do the exact same thing for the other two curves. And what I would show then is that the fitness of W12 relative to W22 is approximately Z times A plus D in that same way. And all of that just followed by basically using this Gaussian approximation. So the reason that we'd like to put this in terms, so this is the difference in fitness, is the difference in the proportion of the individuals that get to reproduce. That's how we're defining our fitness. We want to calculate how the frequencies of these alleles change under selection. So I actually wrote a version of this equation up the last time I spoke, but I'm going to give it to you again here. So under a diploid model of selection with a single locus and two alleles, the change of allele frequency under selection in one generation can be written a few different ways, but here's one way. Already making errors. And if we assume that this is under Hardy-Weinberg, W bar be written in the following way. And these three fitnesses are the relative fitnesses of our three genotypes, and for the model that we're looking at, they're the relative proportions of the populations that get to breed. All right? So I wrote this before. Remember, we wrote it before as PW1, the marginal fitness minus W bar. We've seen it a few different times. This is a slightly extended version. Okay, but we said we can write this in terms of the height of the distribution at that truncation point, and then this A minus D, which gave the width. What about W bar? Well, there's a few different ways to find W bar, but W bar is the mean fitness of the population. Fitness here, we're just defining it as reproduction, right? Who gets to breed. So W bar is just the proportion of individuals that get to breed. That's the mean fitness. The mean fitness is this proportion. So it's this area B in this case. So W bar is B for this case. You can think of it this way. There's a proportion 1 minus B of the population with fitness 0, and a proportion B that has fitness 1. So we can go ahead and replace things. This is going to be Z A minus D. And this is Z A plus D. And this is B. And a little bit of rearranging gives this equation, which is rather famous. So I'll put a box around it to remind me to talk about it. Okay, so this says that under this kind of model, the change in the frequency of the favored allele, the allele that gives the better phenotype, is made up of three things. Thing one, Z over B. And Z over B is an intensity of selection. It's actually, we've seen this before, right? It's that selection differential divided by the variance. So it's the selection differential in terms of the variance of the distribution. Thing two, the allele frequencies. And thing three, the slope of the regression of phenotype on genotype, which I'm so glad I didn't erase. This. I said it was important. This is why. All right? So here we see that it naturally comes out of this very simple model. We have these three things that affect how much this kind of selection can possibly change allele frequency at one focal locus that's affecting phenotype. Depends on how strong you apply selection. That makes sense. It depends on the current allele frequencies, so where the population is. And then it has to do with how much allelic variation there is. I mean, it's kind of ridiculously obvious, but I'll mention it anyway. If either of these are 0, so that the locus is fixed, it doesn't matter how much selection, we're not going anywhere, right? And then finally, this part is the relationship between genotype and phenotype. This is the change in allele frequency. But we also care very much about the change in the phenotype, and I'm just going to focus on the change in mean phenotype. Under selection. We can go ahead and just use our definition of the mean, M, that we found earlier. And then in the next generation, this is going to be this quantity. Do this right? Yeah. Actually, why don't I just do this? P prime minus Q prime A to P prime D. And P prime is going to be P plus delta P, and Q prime is going to be Q plus minus delta P, where delta P is given by this. Now, I've assumed something important. What did I assume in writing these two equations? Kind of a big assumption. Selection is not changing A, it's not changing D. Well, this is a reference point, so that's fine. So it's not changing how this locus is affecting that phenotype. That is a bad assumption to make under really long term, but under short terms, it's probably fine. So if we do this, then we can do a bunch of calculations and find that delta M, just algebra, I don't feel like, we're going to ignore terms like delta P squared. So we're going to assume that the allele frequency isn't changing too fast. And we can then write it this way. Right? And that immediately has, similarity to what's going on with the allele frequencies. This term comes in squared. But now we're going to do something to rewrite that in a way that's more familiar. Let's write Z and B in terms of the distribution of the phenotypes. We said earlier that Z over B was this scaled selection. So we can write that as S over sigma squared. But what is M minus M prime, delta M, in terms of our truncation selection experiment earlier with our phaseolus? Earlier M prime minus M helps if I do it right. The difference between the next generation and the current generation. We call that the response, right? So I can rewrite this way. The response is 2pq divided by sigma squared times this quantity. Too many? No, not too many. There. S. So the Breeder's equation is this. And that tells me right away that in terms of this model, I can define that as the narrow sense heritability. So this is what's usually given as a definition in a quantitative genetics model. It means that the narrow sense heritability looks like this. 2pq sigma squared d minus p quantity squared. Yes? It does have dominance term. So the dominance coefficient enters into both the additive genetic variance and the dominance variance. I'll show that in just a second. Yeah, it actually, D enters into both. It's not that that's not the dominance variance, that's the dominance deviation. So the way this model is defined in the classic sense, D enters into the additive case. You'll see later that epistasis enters into its own variance, enters into the dominance variance, and you have to, yeah. So the dominance deviation does enter into the heritability. And that has to be true, right? Because in that example I showed you that dominance decreased heritability. The dominance actually affects heritability. And it makes sense because it basically, the existence of dominance keeps the effects of the alleles from being completely additive. So it affects, it can increase or decrease. Well, it only really decreases heritability in that case. So if we look at this, you know, and because this tells us how this locus will respond to selection, we can ask the question, when will heritability be very small? When will there be very little response to selection? And you can see that, you know, obviously if there's no genetic variation at the locus, so h squared small, or 0, so either P is 0, Q is 0 for this one locus case, right? Or P1 or Q1, whichever one you want to think about. When the slope of phenotype on genotype is 0, and finally, if this is the phenotypic variation for the trait is large, right? It makes heritability small. That sigma squared is the distribution of the phenotype, the whole phenotype. So if we have a lot of phenotypic variation, and under the rules of this model, our locus affects the phenotype a small amount, then the heritability for this one locus is small. It can't make a big change in the phenotype because it's only a small part of a lot of variation. And the other two things are obvious. If there's no difference in the allele frequencies, we can't select. And then once again, if there's not a relationship between the phenotype and the genotype, or if there's only a very weak relationship, then heritability is small. So all those things are important. Okay. So now we're going to get to what you just asked. That's the mean. Let's think about the variance, because the variance is important. So we're going to assume that the variance for this phenotype for the 3 different genotypes is the same. We assume the mean is shifted, but that the allele is not changing the variance. often, I mean, it sort of depends how you do write this. We often think of ourselves as having a phenotype, and, well, there's a few different ways to write it. Sometimes we imagine there's some overall mean for that phenotype, and then there's some additional change caused by the genotypes, and also caused by the environment, and then possibly change that's caused by interactions between them. If we sort of, set the mean at 0 or not worry about some sort of overall mean, we can also, and I'm going to ignore for now interactions between genotype and environment. Sometimes you'll see this. So the variance then of the phenotype is going to be a genetic variance, an environmental variance, and a function too of this covariance. And we are going to assume this for now. So we're just going to say, we're going to write the phenotypic variance in terms of a genetic variance and an environmental variance. All right? So we can write the genetic variance for our simple one locus example. You can write it out. We can say, well, for just this case where we have a single locus, the genetic variance is going to look like, again, we're assuming Hardy-Weinberg, the mean of the AAs minus the overall mean squared, 2PQ, the mean of the heterozygotes minus the overall mean squared, and finally our other homozygotes, their mean minus overall mean. Right? And we have all those quantities written in terms of our model. One thing that we can do is just rescale this. So we're going to rescale so that our reference point is 0. And that means that our mean phenotype becomes this quantity. Just simplifies things a little bit. And then eventually you can just write this. I keep putting my squared in the wrong place. Okay, that. So, and then the phenotypic variance is that quantity plus some environmental variance that I haven't defined. So these two components of the genetic variance have special names in this model. This component is called the additive genetic variance, written as sigma A squared. And this is called the dominance variance. Or sometimes the dominance component. I don't know. Write it however you like. And this is important, and you brought this out. D appears in both of these. So D has an effect both on the additive genetic variance and on the dominance variance. But we'll see, or actually, we've already seen that the heritability, which determines how our phenotypic distribution can respond to a certain amount of selection, is this quantity. And now looking at that, I can rewrite my heritability this way. where I'm just putting a P here to remind me that that's my phenotypic variance. So we can take the genetic variance and decompose it into two parts. And it's only this part, the additive genetic variance, that determines the response to selection. So, and this is, when I said there was another definition of heritability, this is it. It's the additive genetic variance. It's the part of the total phenotypic variance that's additive genetic variance. Dominance variance does affect the phenotype, but under random mating, which we assume with this Hardy-Weinberg equilibrium, right? We're assuming we're having random mating. Under random mating in diploids and sexual reproduction, that dominance variance doesn't get passed on. Any questions on this part? The dominance variance appears in the denominator. No, this is P. This is the phenotypic variance, the overall variance. I can rewrite that. Maybe I should extend that a little bit. For this simple model where I'm ignoring all kinds of things, it says this. Is that what you mean? That it appears here? Yes, because it's part of the overall phenotypic variance. Additive genetic variance is, it's actually defined as that quantity, but I'm trying to think of like, yeah, so it's the proportion of the genetic variance that determines how selection affects the allele. So it's literally, this is the genetic variance, but this is the part that determines the response to selection, that determines R. It's defined by the heritability, basically. The heritability and the additive genetic variance are sort of intertwined. So this, the handout has a few graphs. Just showing the relationship between these different variances under different cases. So this is the case up here where A is dominant to A prime. And so here's the genetic variance, this curve here, the overall, the total genetic variance. And the curve underneath that is the additive genetic variance. And then this curve is the dominance variance. And you see these curves are flipped when A prime is dominant to A. So this is the case of additive alleles. So when D is 0. So completely additive alleles, all of the genetic variance is additive variance, and there's no dominance variance. So for the additive case, the dominance variance goes away, and everything is additive. And then this odd-looking thing is overdominance, where the heterozygote is the most fit. And that shows something interesting. This is allele frequency, and when you're at equal allele frequencies, the additive genetic variance goes to 0 in that case. And one thing to note about this definition of heritability is notice that this depends on allele frequencies. So the heritability will change as selection changes the allele frequency. And we generally assume that it changes it by a small amount. And actually, that seems to be true. So under selection experiments, where they're selecting on a phenotype, they measure a heritability and then they select another generation. For a few generations, heritability does seem to be constant. But obviously that can't be true forever, because as allele frequency changes, it has to change. And so this graph here shows, so here's heritability, and here's the frequency of the big A allele for the case where big A is dominant, A prime is dominant, and there's no dominance. And you see that heritability is different depending on the value of the allele frequency. The case of no dominance, the case of additivity, is symmetric, and the other cases, of course, are not. So I said that underlying this model was this idea of having many loci. And what we did is we focused on a single locus. So oftentimes, what people do is they do variants of this model. I have a few minutes, so I just can talk about them really quick. Making various assumptions. And they become sort of arbitrarily more complex with arbitrarily more different variance components. But I'll talk about a little bit of an extension here. So we might imagine that we have a single locus, locus 1, and locus 1 has deviations from the overall mean of minus A1, D1, and A1 for the 3 genotypes at that locus, assuming we have 2 alleles at each locus. And we might imagine locus 2 has deviations minus A2, D2, and A2, etc. And when we assume that the loci are generally independent, then what's sometimes called the multifactorial model you can write that the overall change in mean for your phenotypic trait is given by this. How much selection you apply, and then just a sum of the independent effects of the loci. And this assumes that the loci are independent and additive in their effects across loci. And then for any one locus, you can define an additive genetic variance that's just this quantity with the right subscripts and a contribution. So when we talk about these models, there's various degrees of sort of realism or complexity. And I just wanted to sort of, you can write them lots of different ways, but here's kind of a thought for how they get slightly more complex. So the very simplest possible case is the completely additive case. With no dominance. And so it's additive both across loci and within loci. That basically means that each allele adds something independent to that phenotype. And in that case, D is 0. So the dominance variance is 0. The genetic variance is just the additive genetic variance. And the phenotypic variance is just that additive genetic variance plus some environmental component. The slightly more complex case includes dominance, which is the one that we actually spent today deriving. But it's additive across different loci. So now we have both additive and dominance components. And remember that the dominance deviation affects both the additive and the dominance component. And then that case is the one we considered kind of in detail, where we have additive variance and dominance variance. And then the next case includes epistasis. So this is saying we're relaxing additivity within a locus, and this is saying we're relaxing additivity across loci. The problem with epistasis, and I think I'm not even going to draw a cube, a hypercube or anything. The problem with epistasis is it becomes arbitrarily more complex. And so, which is the main reason why I did not deal with it. But in general, you can work out a version of this model which includes an additional variance component that's due to epistasis. We use I for interaction because we're already using E for environment. And because it's arbitrarily more complex, you actually have to define epistasis statistically in your model. I'm going to show a toy example because I think it helps you sort of think about how epistasis might change what we're doing. So this maybe provides a different way of sort of thinking about accounting for epistasis. So let's have the very simplest example we can think of. Two loci, two alleles each. A1 and A2, and B1 and B2. And we're going to simplify matters a lot because I just want to think about how epistasis might affect things. And I'm going to go ahead and set my reference point to 0 and assume no dominance for both. So dA and dB is 0. So there's no dominance at either of the two. So if there's no epistasis, we can write out sort of the mean phenotypes for all these possible combinations a few different ways. I'll do it this way. So let's assume that A, this gives a plus A and minus A, and plus little b and minus little b. So then, and we set m to 0. This is A plus B. This is B. This is minus A plus B. This is A. This is A minus B. 0, minus B, minus A, minus A, minus B. So any deviation from that is epistasis in this format. So let's think of a very simple example of epistasis where there's only one of these two locus genotypes that deviates from this no epistasis case. I'm going to say that the double homozygote A1, A1, B1, B1, has a phenotype, a mean phenotype, given by A plus B plus some additional term, little I. It's different from the additive amount by that much. And so, and we're also going to assume that these are in Hardy-Weinberg. I need Q's. Q1 squared. Yeah, my notation's kind of ugly. P2 squared, 2P1, P2, Q2, Q2 squared. Those are the frequencies. And then these are, this one's A plus B plus I, and everything else is the same. So you can go through the exact same sort of thing that we did before, and assuming that we have Hardy-Weinberg equilibrium, ask, what's the mean phenotype? And we would find that the mean phenotype, after lots of annoying algebra, do I have time? I don't have time. Lots of annoying algebra later, is this. P2 minus Q2 plus I, P1 squared, P2 squared. And this is this interaction term. And if I had instead, if I compare this to the result with a single locus with two alleles, where m was 0, so this is for one locus, we found, letting m equal 0 still, we found this quantity. Wait, and no dominance. Say no dominance. This is the quantity. So I want you to compare these two things. So this is no dominance, and but just this one epistatic interaction. And you see that basically we have a term that's due to one of the loci, and we have a term that's due to the other locus, and then we have an interaction term. So when you go ahead and calculate the variance components for this case, you end up with a term that has I and allele frequencies, a variance term, that ends up, I'm not going to go through it, it's this thing. But in the same way that the dominance deviation showed up in the additive genetic variance and in the dominance variance, right? This is a function of A and D. This is a function of just D. In the epistatic case, this is a function of A and D and I, D and I, and I. So there's a term that just has this interaction component, but it ends up in the same way that the dominance component comes in. So it becomes sort of arbitrarily complicated. So I just want to say that this is sort of a statistical way of thinking about epistasis. How does it shift the mean? And how does it shift the variance? When people who work on quantitative genetics in either experimental populations or in natural populations try to figure out epistatic interactions, that's how they do it. They look at the means and variances of various kinds of individuals and calculate it statistically. Then the question is, well, how do you go from that to actually finding what the actual gene interactions are? And they usually use some sort of like focal loci and do sort of breeding experiments. So it's not at all straightforward. There's a lab at Kansas, John Kelly's lab, that works on Mimulus cutatus, which is the yellow monkey flower. And they have some very well-characterized wild populations where they've isolated, over many years, a set of loci that have fitness effects, strong fitness effects, both on the size of flower and on the first day that these plants flower, which is important because these plants live in high elevation, so they only have so much time to flower and set seed. And so they have a set of loci that affect these traits in the greenhouse and also in nature. And Patrick Monahan, who was a former grad student of John Kelly's, and John, and then Patrick later on his own as well, did a whole bunch of field studies where they looked at crosses that either had one of these mutations, had another of these mutations, or had the double mutant, and looked for epistatic interactions. And in some cases they found them, in some cases they didn't. But it's very hard to find them in natural, it's very hard to characterize them in natural populations because you're looking for variance. So you're looking for variance above and beyond the kinds of variances that you'd see naturally. Okay, I'll pick up sort of from this, and then we'll talk about how natural selection affects phenotypic traits next time tomorrow. But I think I could answer a question or two. If there's one. Everyone's like, pretty straightforward. All right, thank you.